Welcome back, guys. This is part 4 in our introduction to biological molecules. In the previous videos, we went through carbohydrates and lipids and proteins, and now we're on to nucleic acids. So just as a reminder, we finished the last video talking about protein structure, and proteins are made of amino acids to build complex structures that we call proteins. And the next section on nucleic acids is especially important because the sequence of those amino acids that we use to make a protein, the cell has to have instructions to know what sequence of amino acids it's supposed to use. And those instructions are entirely given by nucleic acids, which means DNA and RNA. Okay, and so when we talk about DNA and chromosomes and genetic information, we're really talking about these molecules called nucleic acids. And what they're made out of is really 3 things. You have a phosphate group, this PO4. You have a sugar, which is this pentagon-shaped molecule called ribose. And then you have a base, right? And so phosphate, sugar, and base. The phosphate and the sugar really never change. They're the same in every nucleotide. We call the monomers, this entire thing together is one nucleotide, right? That's what we use to make chromosomes to make your DNA out of. The base is the part that's variable. Okay, so these molecules up here, you divide them into 2 categories. Pyrimidines include cytosine, thymine, and uracil. Cytosine and thymine are only found in DNA. If you are working with RNA, which is a very, very similar molecule, but the sugar is just slightly different, we use C and U instead of T. Right? And purines, the other 2 types, A and G are purines. They're used for both DNA and RNA, and there's really no difference. Right? So the only real variable that we've got here is what base are we using and if it's RNA or DNA. The difference between RNA and DNA entirely comes down to whether or not there are these OH alcohol groups on the carbons in the molecule. So if the carbons have 2 OH groups, it's RNA, just ribonucleic acid. If you're missing this OH that would otherwise be here, we say that that's DNA. The D is for deoxyribonucleic acid, right? Deoxy meaning we don't have an oxygen coming off of that carbon. Right? Otherwise, incredibly similar. All right. When you start putting those DNA nucleotides together to form a chain or a polymer, we just call it DNA. And so this double helix structure that I'm sure you've seen before that makes up chromosomes, if you zoom in on that, each one of these molecules that you're seeing, each one of those is one nucleotide, right? And we're linking nucleotides together in 2 different ways. The first is you're linking them together to make one strand of DNA. So we say that this double helix is double-stranded, right? If you were writing it out, that would be just linking together DNA nucleotides like this in a chain to make a polymer, just like linking amino acids together to make a protein. Now, the double-stranded part of it is telling you that the 2 strands will stick to each other. And the rule is A's always bind with T's, which means T's bind with A's, G's connect to C's, and C's connect to G's. And so every double-stranded piece of DNA is really just 2 strands, 2 separate polymers that have hydrogen bonds holding them together in the middle, right? And this is what it ends up looking like. So if we did a close zoom in on 2 separate base pairs, what you would see is nucleotides, right? That is one thymine nucleotide. This is a guanine nucleotide. This is an adenine or an A, and this is a cytosine or a G. So we have 4 separate nucleotides there, right? Each one has a base, it has a sugar, the deoxyribose, and it's got a phosphate. Right over here, this would be the base, that's the sugar, that's the phosphate. And so for each of these, they're held together by 2 different types of bonds, right? The bonds that are holding together the backbone, right? That's this. The sugar and the phosphate make what we call the backbone of the DNA strand. And this bond with the phosphate that holds those nucleotides together in their backbone, we call that a phosphodiester bond, okay? So phospho because there's a phosphate, diester because there are two oxygens, right? So phosphodiester bonds are covalent bonds that hold one strand together. These dashed lines that you're seeing that are causing base pairing, those are hydrogen bonds, right? So if you heat DNA hot enough, those hydrogen bonds fall apart readily and split. So you can peel the two strands of DNA apart from each other by breaking those hydrogen bonds. The phosphodiester bonds are only cut with enzymes. Even if you heat the molecule, those are very stable because they're covalent, right? The only other difference to maybe pay attention to is the hydrogen bonding structure. Adenine and thymine have two hydrogen bonds, right? 1, 2 that link them to each other. G and C, guanine and cytosine, have 3 hydrogen bonds that bind them together. That means that G and C stick more tightly to each other. They're harder to peel apart. You have to add more heat and increase the temperature in order to get them to separate. A and T, they peel apart a little bit more easily. Those hydrogen bonding differences are also what allow us to use the base pairing rules. The enzymes that make DNA in your cell, they know that A and T go together because it's like a puzzle piece. Their hydrogen bonds click together. G and C go together because theirs are a match. And if you tried to pair C with T or G with A, the hydrogen bonds wouldn't line up, and you would have a distortion in the double helix, wouldn't have the right shape, and repair enzymes in your cells can detect that and see that there's a problem. Okay. All right, the last thing that I want to do with this is link it to future directions in the class. So in putting this whole DNA protein thing together, what I want you to see is this is the most fundamental concept in genetics. Your DNA in a eukaryotic cell is found in the nucleus, and consider the DNA like a permanent copy of information, almost like it's being stored in a library. When you go through a process called transcription, which is going to be incredibly important later on in this class, transcription just means that you're making a temporary copy of your DNA in the form of RNA. The difference is DNA is double-stranded, like we just looked at. RNA is single-stranded. And we're not turning the DNA into RNA. We're making an RNA copy by reading the DNA. And so think of it more like making a Xerox copy of a scanner, like for a book, right? So if you take a book and you want to take copies home with you, but the library won't let you, you throw it on a Xerox machine or a scanner and print out a copy. The RNA is kind of like that temporary printed copy. And the RNA gets to leave the nucleus, head to the cytoplasm, and by translation, right? Translation is what ribosomes do. They convert that mRNA sequence into a polypeptide, and polypeptide is really just a synonym for protein. Each one of these little dots on the polypeptide is representing one amino acid. So to link all of these biological molecules together, you're using your sequence of DNA to make an RNA. Those are both nucleic acids. The RNA leaves the nucleus and tells a ribosome what sequence of amino acids to use to make a protein, right? That protein can serve lots of functions. Some proteins are structural. Some of them act as enzymes, right? And those enzymes might be used to go metabolize nutrients that give you energy for your cell, right? In the form of things like carbohydrates or lipids that we saw in the earlier biomolecule section, right? So carbs and lipids are mostly an energy source. Proteins serve all sorts of different functions that actually carry out specific activities inside of a cell. And your nucleic acids, DNA and RNA, are just what tell the cell what proteins to make. Okay, so I hope this sort of ties it all together and gives you an overview. The next section in this playlist is going to go towards basic cell biology, looking at the structure of cells and organelles. And that's pretty soon going to eventually lead into a discussion of genetics. And so all of these ideas are going to tie into each other. Make sure you bookmark the playlist, subscribe to the channel. There are going to be a lot more videos to come, and we'll walk you through all these topics to clear up any confusion that you might have on future chapters.